Hello, everyone. Welcome to today's live broadcast, Amorphous Dispersion Drug Formulations, a modern simulation-based approach. I'm Rita Peters, the Editorial Director of Pharmaceutical Technology, your moderator for today's event. We're pleased to bring you this educational web seminar presented by Pharmaceutical Technology and sponsored by Patheon. Patheon is a leading global provider of pharmaceutical development and manufacturing services. With approximately 8,000 employees across 26 locations around the world, Patheon provides a comprehensive, integrated, and highly customizable set of solutions to help customers of all sizes satisfy complex development and manufacturing needs at any stage of the pharmaceutical development cycle. To learn more, please visit www.patheon.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. If you'd like a copy of the presentation, submit your request using the Q&A box. I'd like to now introduce today's speakers, Tom Reynolds and Matt Wetzel. The first presenter, Tom Reynolds, has more than 20 years of experience in research, development, and management of programs in nanotechnology, formulation, crystal growth, and computer modeling. In his role as principal scientist in the Global Sciences Division at Patheon, he uses a computational-based technology platform to identify and develop drug formulation and solubilization options for clients. Tom earned his doctorate in chemistry from Oregon State University. Matt Wetzel, a principal scientist in the Global Science Division, has 20 years of experience in the pharmaceutical industry as a computational chemist. In his current role, he's primarily responsible for developing predictive computational approaches that address challenging solubility and formulation problems. Matt spent 10 formative years at Pfizer working as a modeler on several drug discovery projects. Along the way, he earned invaluable experience in medicinal chemistry, formulation science, and information technology. Prior to joining Patheon, Matt worked for Bed Research and was a solutions architect at Schrödinger. This combination of skills and experience allows Matt to impact a multitude of areas within Patheon and uniquely places him at the forefront of the company's predictive modeling efforts. He holds a bachelor's degree in chemistry from Washington State University and earned a doctorate in chemistry from the Pennsylvania State University. Thank you both for joining us today, and Tom, you have the floor.
Okay, thank you, Rita. Hi, everybody. As you can see, our title of our talk, Amorphous Dispersion Drug Formulations, a modern simulation-based approach. What we are going to talk about is a platform that we have been developing at Patheon to predict what formulations will be used in solubilizing poorly soluble drugs. So on this slide, what we see, we typically get a crystalline drug, poorly soluble, and to increase the solubility of this, we want to mix this with an amorphous polymer. And this can be done by co-mixing or dissolving in a solvent, hot melt extrusion, or spray dry. And ultimately what we want to have is an amorphous dispersion where you have the drug molecules in the red molecularly dispersed in the polymer. And as I can see on the right-hand side of the screen, I have an amorphous dispersion. Its energy is somewhat higher than the crystalline drug, and also somewhere in between the crystalline drug and the amorphous drug energy. So the stability of this amorphous dispersion is only as good as the thermodynamics. So if we have a stable amorphous dispersion. We expect good stability and perhaps good dissolution behavior. What we do at Patheon here is we'll take, for example, a 2D structure of a drug, dipyridamole, shown in the upper left-hand corner. We do a whole series of quantum calculations and come up with a range of molecular properties. And of course, we look at a variety of drug conformations, and then what we do, if I go down to the lower row, is I look at a variety of different descriptors, and the simplest ones that we're all familiar with will be things like hydrogen bond acceptor, shown in the green, hydrogen bond donor acceptor, shown in kind of the yellow color, aromatic and hydrophobic. This is just one set of descriptors that we like to look at. We also look at a variety of surfaces. And here is an electrostatic surface derived from a quantum calculation. And we've looked at this through density functional theory at a fairly high level. And what we can see is the molecule is got a variety of different charges to it. It's got a positive surface down in blue, red surface down in back, and neutral, which would be more of a hydrophobic or aromatic surface, in green. So what we're really looking for is what I would call the sticky parts of the molecule. How is this molecule going to interact with itself in the crystal structure? And more importantly, how will this molecule interact with different excipients? And so we come up with a variety of these different descriptors. And then, as you can imagine, what we want to do is find in an excipient a complementary set of descriptors. So we do the same type of calculations on numerous different excipients. These are typically polymeric, and I've shown just a block structure here of PVAP, polyvinyl acetate phthalate. And you can see on the right-hand side of the screen, we've got the same set of hydrogen bond doning, hydrogen bond accepting, aromatic, and hydrophobic descriptor type groups. So the idea is to come up and match the drug with an appropriate excipient based on complementary surfaces and descriptors. Now, as you can imagine, these different descriptors based on both change, the amount of them change upon drug loading, other additives that might be included in the formulation. So, for example, with dipyridamole, if I take a look at lead polymer candidates, and what I've done is screened probably 20 different polymers and then come out with a lead case of polymers. And so these would be something like HPMCP, HPMCAS, PVAP, PVPVA, and Solu+. So then we interact the drug molecule and the polymer molecule together in molecular dynamic simulations, and we find out what these interaction energies are. And so what we have in this slide would be the solid state of the drug and polymer interaction. And this is very akin to what you would find in a drug development program, where you have the drug molecule shown in the stick figure and the polymer shown in the space-filling model, the balloon-type model. And what we see is the drug is binding in pockets. And these are largely, what we have found, largely places where the complementary descriptors interact with each other. And these binding energies can be anywhere from 10 kilojoules per mole to 100 kilojoules. Now, in these simulations, we're dealing with anywhere from 10 to 50 drug molecules in these small-scale molecular dynamics. When we want to go to something larger, where we have something like dissolution in water, we need to go to a high-performance computing platform. And Matt, I'll let you take it from there.
Okay, Tom, thanks very much. So, as Tom was saying, initially we will run simulations of anywhere from 1 to 10 drug molecules and a few polymers. But as you see in the middle, we can actually, with our high-performance computing platform, we can run hundreds of drug molecules and polymer molecules at the same time. Typically, you have between 1 and 10 drug molecules and some polymers that will run in the simulation locally. But when we want to get more accurate energy estimates, we want to sample over a large time and space and even deal with solvent effects or other excipients that might be included in the dispersion, we'll actually need to go to a high-performance computing platform where we can actually model hundreds of drug molecules and polymer molecules at the same time. So these are now simulations of tens of thousands or even hundreds of thousands of atoms. And we run these on a high-performance platform using Amazon Web Services Cloud, which you'll see here on the next slide. So the actual setup that we run is using a software system called LAMPS, which is freeware. It was developed at the Sandia National Labs down in New Mexico. The force field that we use is the Merck molecular force field, the static version, and the system sizes, as I said, is anywhere between tens to hundreds of thousands of atoms. Now, we run these simulations using high-performance computing platform Linux systems on AWS Cloud. And so essentially we're renting time, CPU time, from the AWS Cloud. And that allows us to cut down on costs because we don't have to actually have our own internal hardware. So if you look at the image on the left, you've got dipyridamole, which is the drug that Tom was talking about, in the green surfaces, and PVPVA64, polyvinyl pyrrolidone vinyl acetate, are the gray sticks. And this is the result of a simulation run where what we're seeing is the result of a simulation where we see some agglomeration of drug molecules and some drug molecules that are actually separated and individually placed within the lattice of the polyvinyl pyrrolidone vinyl acetate that you see there in the gray sticks. Now, the PVPVA is not a crystal, obviously. It's an amorphous, and the entire system is an amorphous dispersion. And so what we're looking to understand is, first of all, what are the interaction energies between the green blobs of drug molecule and the gray sticks, or the polyvinyl pyrrolidone vinyl acetate, as well as what is the agglomeration. So if you look at the upper right, the top right, or the top portion of the image, you can see a quite large green surface. Well, this is agglomeration of drug molecules that we actually see in the simulation. And what this tells us is that these are potential nucleation sites for growth, for crystal growth. So over time, this might lead to instability of the dispersion. So we're looking for situations where we have incompatibility between the drug and the polymer, which leads to these agglomeration sites. So before we really get into the simulation elements and the results that we see from the simulations, I want to take you through what's actually happening in a solid dispersion. Regardless of how you got to that point, whether it's from a spray dry or from a hot melt extrusion or from some other technology, and there are others that are available for this sort of thing, essentially what you need to do is create a homogeneous mixture of your drug and your polymer. So initially what you can look at is a physical mixture of pure crystalline drug and the polymer of choice. In this case, we're talking about nifedipine. That's what the NIF stands for. So that's nifedipine is the drug, and PVP is just polyvinyl pyrrolidone. Initially, we take the nifedipine crystal and the polyvinyl pyrrolidone powder, and we crush them up with mortar and pestle, if you will, and make a physical mixture of them. But they're not intimately mixed at that point. You still got microcrystals or nanocrystals of nifedipine in a physical mixture with PVP. Then what you do is you add heat to the system. And what you see is the heat of fusion, and that's for the drug molecule. So as you add heat to the system, what's going to happen is that the drug molecule, which is a crystal, is going to actually melt. And it takes the heat of fusion to do that. So you have to add a certain amount of heat into the system in order to get the nifedipine crystal to melt. Before you get to the melting point, you're obviously going to raise the temperature of the entire system, so that's additional heat that you're adding. But once you get to that heat of fusion point, the melting point of the crystal, you have to add in the heat of fusion in order to melt the crystal entirely. Once you melt the crystal, then you've got liquid nifedipine crystal at an elevated temperature and PVP at an elevated temperature, and that's when they begin to intimately mix. Now, when they mix, there could be heat released or there could be heat absorbed, and that's actually what we can try to measure. So if the amount of energy required to actually melt the crystal and then form an intimate solution is less than the actual heat of fusion, then there was some exothermic reaction and there was heat released upon mixing. And that's what you see here, is that there could have been a negative delta H mixing, a release of heat, to give you this mixed system, which means that when nifedipine as a liquid and PVP as a powder are mixed, that's a favorable mixing that's occurring, at least enthalpically speaking. There's also entropy to consider, but we're not going to get into that in this presentation. So then you're left with a liquid solution of PVP and nifedipine, and you can actually measure the energy it takes to go from the ground state, which is just the physical mixture, up to the mixed state. And if that's different than the ideal amount of heat it would require, which is based on the heat of fusion, we can actually look at the difference and call that the heat of mixing. And that's illustrated much more clearly on the next slide. Here you have the heat of mixing physical situation. So the energy, which actually is endothermic up, you see that you've got crystalline nifedipine at the base of the low baseline. Slightly above that would be if you had amorphous nifedipine, which you could have as well. You could make amorphous nifedipine and add that to your PVP. Well, obviously that's not going to have a melting point per se, but there would be some heat required in order to get that to become a liquid. But let's just take the crystalline case. So you see that the crystalline nifedipine, you're going to add energy to that to melt that crystalline nifedipine. The amount of energy needed is the heat of fusion, as you see. And you get up to the dotted line, and that's actually the amount of energy it takes to melt your solid nifedipine in the crystal form and get it into a liquid. But then what might happen is that you actually see a decrease in the overall energy state, because as they mix, energy could be released, and that would be an exothermic event. Now, what we can actually measure using differential scanning calorimetry is we can measure the energy required to go from the ground state, which is crystalline nifedipine, PVP, up to where you see the liquid solution of PVP and nifedipine. We don't measure the energy it takes to get to the dotted line. We measure the energy it takes to get from the lower solid line to the upper third solid line, which you see is the yellow box is pointing to. So that's actually the energy that we can measure experimentally, and that's the energy that we care about when we're running simulations, because we want to try and incorporate simulations that actually predict what that energy change is actually going to be. So in other words, that can actually derive directly the heat of mixing upon integral mixing of the drug and the polymer in an actual dispersion. Now, it may be the case that in some systems you actually see an endothermic event where upon mixing you have to actually add in more energy to get the system to mix. Well, when that happens, you probably have a situation where you have an unstable dispersion that's going to just phase separate and recrystallize, at least the drug part of it will recrystallize into domains, and then you've effectively defeated the purpose. And we can measure that too, and we can look at that from a simulation perspective to try to understand that this polymer drug combination is not going to make an effective dispersion. Okay, a little bit more complex math here. It's all not that complex, but I'll go through it quickly. So if you look at the enthalpy of mixing, what you see here is a plot, and it's endo up. So what you see is a plot of the heat of mixing, or the enthalpy of mixing, as a function of drug loading, which is phi sub d. So the phi here is just the amount of drug in the system. As you see, as you go from 0 or 0% drug all the way to 100% drug over to the right where it says 1, you can see that your heat of mixing is going to take on this parabolic shape. So somewhere in the middle is where you're going to have your maximal heat of mixing. Obviously, if there's no drug, there is no heat of mixing. And likewise, if there's no polymer, where drug is 100%, there's also no heat of mixing. Therefore, you expect those values to be at 0 on the y-axis. Now, what you can actually do is you can actually measure through some calculus. You can just derive the slope of that function form, and you can actually look at the heat of mixing that each individual component is contributing to the system. And this allows us to understand the non-ideal behavior of the system. Now think about it in these terms. If you take 50 milliliters of ethanol and 50 milliliters of water and you mix them, you get two things back out. You get heat and you get a change in volume. The final result of 50 milliliters of ethanol and 50 milliliters of water is about 96 milliliters. So you've lost some volume. That's because the ethanol and the water mix and form tighter bonds with each other, and so the volume they contain actually decreases. And you actually see a release in heat too. And that's what we're trying to measure here in a solid solution of drug and polymer. Now when we do that, what we actually see is demonstrated in this slide here. So this is now some real experimental data that we've actually measured using nifedipine and PVP and looked at the enthalpy of mixing. So we've actually used differential scanning calorimetry and then calculated the ideal mixing, heat of mixing, as if both components didn't actually mix. And then we look at the non-ideal behavior. So what's the difference between what we measure and what we expect? And what we see is a similar type of shape as in the previous slide, which I just showed here. I'll go back to real quick. You see this parabolic shape. Well, in this slide, you also see somewhat of a parabolic shape. And we believe that these points out here, where we've got the red ellipse, are areas where the amount of drug that we've actually added to the polymer is very high and the amount of polymer added is low. So we might have some inconsistencies in our measurement in terms of the weight fraction, the mass fraction of drug and polymer, and so we think these might be slightly in error. But essentially we do see that this system starts at 0 and then ends at 0 as you get to 100% nifedipine. So this is what we're trying to reproduce from a computer simulation perspective, because these are very difficult experiments to actually put together and do. And we wouldn't want to do them as a way of gauging the quality of the dispersion over time. We would rather simulate these in the computer so that we can then make intelligent decisions about which systems we want to go forward with. Now, I might add at this point that not only do we do this with this type of system, but we can do this with other systems as well. We could do this with lipid systems. And in fact, this leads into the fact that we have a way of understanding from your drug molecule what the best technology is to go forward with. And then once we understand what the right technology is, we can apply additional modeling of the form we're talking about here in order to pick which excipients are optimal if you're talking about, say, a spray-dried dispersion formulation or a hot melt extrusion formulation. So enough math. Get back into some pictures. So if you look at this slide, I just want to remind you that what we're actually trying to do is simulate what you see in the right-hand box. Can we make a system of drug and polymer, have that system mixed in a molecular dynamics simulation, and then from that extract useful information? So let's actually get into whether we can do that. So here on this slide, what you see is nifedipine and PVP after a simulation has been run. So again, I'm taking that solid solution of PVP and nifedipine where I've taken the crystal, I've melted it, and then add that to the PVP, and I form this intimate mixture, this dispersion. Well, let's simulate that by placing lots of different polymer molecules and drug molecules together and letting the molecular dynamics simulation run to intimately mix them. And what you see in the middle is the result of that. So this is a 10% by weight loaded dispersion of nifedipine and PVP. And you can see that in red on the surface is the polymer, the PVP, and then the green blobs are nifedipine. And for the most part, you've got what I would call a pretty good dispersion because you see that there's an individual drug molecule here and there that are well separated from each other. And that's exactly what you want in a dispersion. You don't want agglomeration of drug molecules. You want the drug molecules to be dispersed completely within the polymer matrix so that when this system goes into water, it dissolves and the drug is released as individual drug molecules so that there's no chance of nucleation and you can achieve a supersaturated state of your drug molecule in water. And that's exactly what we're looking for. Now, the density of this system was calculated to be 1.062, and that's actually very close to the actual density that we can measure for this, and you can measure this with a lot of different methods. Stereopycnometry is one method that you might choose. But the density that we've calculated is very close to the actual density that we've actually measured for this system. And so this tells us that not only are we simulating the system accurately, but that the charge model that we're using and the force field that we're using and the molecular dynamics engine that we're using is leading us to results that map to what we see experimentally, which tells us that we can take this forward into systems where we don't have experimental data so that we can extrapolate and predict on new systems. So we can take any drug molecule, any polymer system, run these simulations at different loadings, and we can get interesting information out that tells us about not only the stability of these dispersions, but actually what the performance might be for these dispersions as well. Now, there's more to this than just the pretty picture. We also look at the actual information derived when we run these simulations. So on the next slide, and I'll take some time to go through this, you see different loadings of drug and polymer. Now, the first thing to notice is as you go from the left to the right, you've got 10% nifedipine, 20% in the middle, and 30% by weight nifedipine in PVP. Now, the 10% looks very similar to the previous slide where you've essentially got a true dispersion. You've got drugs that are basically individually spaced within the polymer matrix, and this is all an amorphous matrix, such that the drugs would be released very, very rapidly and effectively. The 20%, you can start to see that while you still have primarily drug that is very well dispersed, you've got some situations where you've got maybe one or two drugs that are close to each other. Now, if we were to change the drug from nifedipine to another drug like dipyridamole, you might actually see a decrease or an increase. We don't know. We'd have to run the simulation, but you might see a decrease or an increase in the amount of drug that is agglomerating. And that has to do with the, like Tom was saying, with the complementarity between the polymer and the drug. The polymer and the drug might not be very complementary. They might not have very good complementarity. So the drug might actually have electrostatic surfaces that are oppositional to the electrostatic surfaces of the polymer, and in that case you might see agglomeration of drug. Or you might have very intimate mixing of the drug and polymer because they are very complementary in terms of their surfaces and shapes. Now, if you look at the 30%, you start to see even more. So there's two things that are happening here. One is that the nature of the drug and the nature of the polymer is going to be one of the determining factors in whether you see agglomeration. But the other is, of course, how much drug is there. So when you have a large amount of drug, say 30%, which is actually starting to become a fairly large amount of drug in the system by weight, you actually start to see agglomeration. And we see this with a lot of drugs. This is why a lot of dispersions really can't push beyond 30 to 40% loading. And that's just because of the nature of the statistical mechanics of the system. If you think about a ball, if you think about a jar full of marbles, some are green and some are red, as you increase the number of green marbles, you're going to see more cases where green marbles are next to each other. That's just by the nature of statistical random mechanics. But the extent to which we see this can also be modified by the nature of the drug molecule and how it interacts with the polymer. And so as we run these simulations with different drug-polymer combinations, we see this breakpoint occur at different places. And that's really what tells us what our loading maximums are likely to be and how much drug-polymer system we can actually make and where we should start our experiments. Now, the other thing to mention here is just because you have drug molecules that are agglomerating together does not necessarily mean that that's bad from a performance perspective. Now, we don't want to see agglomerates because that can lead to nucleation sites and recrystallization. However, if the mechanics of that are incredibly slow, which they typically are in a dispersion, then that might be okay. And in fact, if you have agglomeration sites but still amorphous in nature, so the drug is not actually crystalline, it's amorphous. Well, when those are released into water upon submersing the entire dispersion in an aqueous environment, you still would see very good performance. And that's what we're aiming for. Now, if you look at the bottom two plots, these plots show, on the left, density as a function of loading. So at 0 loading, the density is the density of the polymer. And at 100% loading, out at 1, the density is just the density of the drug. What you see is what you expect, a linear increase in density as you increase the amount of drug. Basically, it starts to approach the density of the drug as the loading approaches 100%. What's interesting is that what we can do is extract out what we call the pair energy, and this is the non-bonded energy between all pairs of atoms in the simulation in joules per gram. And this is derived from Leonard-Jones potentials or dispersion forces, as well as Coulombic interactions. So if an atom is charged positively and another one near it is charged negatively, that's a positive interaction from an energy perspective. What we can do is we can calculate this energy directly from the simulation, and you see that you've got this non-ideal behavior. This is exactly what we looked at from an experimental perspective. You've got excess energy. It's endothermic in this sense. So you're seeing here, essentially, the pair energy is modeling the enthalpy of mixing. That's directly from the simulation. You see this very nice plot here that I'll actually blow up on the next slide that shows that we're modeling this non-ideal behavior that we measured experimentally for the exact same system, nifedipine and PVP. So moving on to the next slide, what you see here is a comparison of nifedipine and PVP from the molecular dynamics simulation on the left and the experimental data that I showed earlier on the right. Now I'm doing, it's XO down, so the negative values are actually where you would expect them to be. I've just flipped the graph. Now you see the simulated data. At 0 loading, you've got no enthalpy of mixing, and at 100% loading, you've got no enthalpy of mixing. But in between, you see an exothermic enthalpy of mixing in the simulated information. And the blue line and the red line are just the differential heats of solution for nifedipine in red and PVP in blue. And what we care about mostly is the infinite heat of dilution the infinite heat of solution at infinite dilution. So for nifedipine, it's around -31, and for PVP, it's around -40. You compare that to what we calculated experimentally, we've got -58 for nifedipine and -127 for PVP. So the energy values are not quite the same, but they're trending in the right direction, and we think we have information and understanding as to why we don't actually generate the same energies from the simulation as the experimental. But what's important is that we get the exact same behavior, and we know that we can tweak these simulations to try to further validate with the experimental data that we have. So we've got a very good model in place that can tell us that when we take nifedipine and PVP and mix them at all weight fractions, we're going to get an exothermic heat of mixing. Now, the free energy has to do with the entropy, and we can calculate that as well. We're just not going to cover that today, because ultimately what you really care about is the free energy of mixing. And if the free energy of mixing is actually negative, you know you've got, in theory at least, an infinitely stable dispersion as long as you keep it away from any other elements in the environment. So on its own, in a vacuum, that would be an infinitely stable dispersion if it had a negative free energy. Obviously, that's not always the case. You might have humidity, you might have impurities in the system, and that could lead to phase separation over time. But what you're looking for is good stability, and then hopefully that can draw you into understanding performance as well. All right, so moving forward, what you see here is just a look inside what these dispersions actually look like. So on the left is just the surface of a brick of the PVP and nifedipine, and then in the middle I'm starting to cut away, and then you can see on the right that the green blobs with a little bits of orange are the actual drug molecules, and this is about a 40% loaded situation here. Or the green blobs are the drug molecule, and then you can kind of see the lighter bluish color in sticks are the actual polymer. And what you see is a very, very homogeneous system. And I might add that spray-dry dispersions produce this homogeneous system by and large most of the time. The reason for that is because we're already taking the polymer and the drug prior to making the dispersion, and we're mixing it intimately in an organic solvent, like, say, acetone or tetrahydrofuran or ethanol. And so by nature, you've already created, quote, a dispersion with three components: the solvent, the polymer, and the drug. And then when you rapidly evaporate the drug, you're maintaining that homogeneous structure in your dispersion. That might not always be the case with hot melt extrusion or other technologies that don't rely on the intimate mixing of a solvent. And so this might be one of the advantages that we see with spray-dry dispersions, is that we actually do produce this very homogeneous system. And in fact, that's why sometimes you see differences in behavior, dissolution performance, between a spray-dry dispersion of the same materials and a hot melt extrusion of the same materials. We believe that might be the case. What might be going on there, I should say, is that you've actually got a non-homogeneous system in the hot melt extrusion, so it behaves differently. It's literally a different material than the homogeneous system of the spray-dry dispersion. So that helps us understand stability, and again, we use this technology to help understand which excipients are going to perform best with a particular drug molecule of interest, and we also use this to help our clients select the technologies of interest that are going to be most effective, because it might be that a lipid formulation works better. And in fact, these simulations can be extended to just about any material that you can get the structure of. If you know the structure of the drug and the structure of the material that you want to make your formulation with, we can do these simulations to try to understand the thermodynamics of those situations. So moving on to the next slide. We've shown that we've got this very useful approach for predicting the stability and understanding the thermodynamic behavior of these dispersions. What about dissolution? Well, that's where we're headed next. So we've covered stability, and what we're going to focus on in the future with our research and development endeavors here at Patheon is actually looking at the dissolution. And we think that there are ways to do that by looking at surface kinetics of these dispersions, and then when you add water in a simulation, how rapidly do things become dissolved in the water? So how rapidly do surface phenomena occur, and can that be correlated with known dissolution performance? But when you think about the dissolution of a drug in a dispersion, this is what's actually happening. Got your dispersion particle, which you see here in the yellow box with the red balls in it and the squiggly lines, the spaghetti and meatballs picture of a dispersion. You add water to that, and what happens is you're going to release polymer into the water as well as drug bound to the polymer. So you're going to create some species that are drug bound to polymer in solution. You're going to create some supersaturated free drug in the middle, and that's what you ultimately care about, is the degree and the extent to which you have supersaturation. But what happens is you've got these competing forces, and the green arrows and the red arrows are basically the rates of change between those different species. So in the lower left, you start to see amorphous drug precipitates, and this always happens. The extent to which this happens is what we're trying to minimize. But it does typically always happen. And you also see crystallization of drug. You see drugs start to precipitate out as crystalline too. And you see interconversion between the amorphous and the crystalline. But by modifying the nature of the loading of the dispersion, the loading of the drug in the dispersion, and two, the nature of the polymer and how it maintains the drug in solution, you can enhance the supersaturation and inhibit the crystallization and the amorphous. And there's a third sink to the drug as well, and that's the intestinal lumen. So what you want to see is the supersaturated drug then absorbed into the system, and that's actually how dispersions that work actually perform. They create this supersaturated state. The polymer maintains the drug in solution, inhibits precipitation, inhibits amorphous drug precipitates, and allows for free transfer of the supersaturated drug into the intestine through the lumen in the jejunum and the duodenum of the body. And that allows for an enhanced solubility, which allows for an enhanced permeability and bioavailability of your drug in the system. Okay. So in conclusion, what we've talked about here is using high-performance computing to model what happens when you take a drug and a polymer, in this case nifedipine and PVPVA. You can do small-scale simulations that we do, as Tom had talked about, to understand what excipients are going to work best, and then you can do large-scale simulations that you see in the upper right corner to extract useful information about the thermodynamics of the system, which allows us to then predict what the stability of the system is going to be, and then ultimately we're going to move forward and talk about the performance of these systems as well. So we've got a very powerful platform in place that helps us derive this information and help make intelligent decisions early in the process to guide our clients as to what the best formulation is actually going to be moving forward. And that's what the Quadrant 2 technology that we have here at Patheon is really all about. It's going from structure, a 2D structure, to a 3D simulation, to useful information about the energetics of the system that help us understand stability and performance and choice of excipients moving forward. And with that, I'd like to finish and acknowledge Marshall Crew, who has helped us along the way and was actually the founder of Aegir Pharmaceuticals, which was recently acquired by Patheon, as well as Sanjay Kanagurthu, who's also in our group and who helped us with a lot of guidance going forward for our group and for the research and development endeavors that we've actually undertaken. And with that, I'd like to conclude, and we'll move into the question and answer session.
Well, thank you very much, Matt and Tom. We are ready for Q&A. Just a reminder to the audience to submit your questions. You can type them in the Q&A box, which can be found on the right side of the presentation window. So here is our first question. In your MD simulation, are you starting from a solution of a drug or a solid?
So in the MD simulation, we're actually starting from a solid drug and a solid polymer, and we're just taking the drug and the polymer and we're interspersing it in a lattice, and then we allow the simulation to run from there. So we essentially run the simulation at a high temperature and cool the system down, and then we look at the energetics of the cooled system once it's actually condensed and given us a density that makes sense. So we don't actually start from a solution of the drug, we actually start from solid drug, but it's really individual drug molecules dispersed inside of a polymer matrix to begin with.
Okay, thank you. Here's our next question. So during the simulation activity, does the number of polymer excipients selected exceed the number of active drugs? So is it not a 1-to-1 ratio?
Yeah, in this case, we can have a variety of drug loading, so it's not necessarily 1-to-1. We can go from 1 weight percent drug loading all the way to 100 weight percent drug loading. So this varies, and we vary them systematically when we do these types of calculations, and it's actually very helpful to look at that because what we can do is arrive at a maximum drug loading for a given excipient using those type of techniques by varying drug and polymer ratio.
All right, thank you.
Yeah, and so
go ahead, Matt.
I was going to follow on to that. So like Tom said, and we do this by weight. So the weight fraction of drug and polymer ranges anywhere from 0 to 100%. Now, if you actually convert that into moles, then it's different because a mole of polymer is there's a lot fewer moles of polymers in a certain weight of polymer than moles of drugs. And so if you look at this from a mole perspective, that changes everything. So we don't do that because everything is squished up to the right. So what we actually do is look at these things from a mass perspective, because it's easier to understand that, and it actually spreads out between 0 and 100% more effectively.
Okay, and we have another question about the API and polymer ratios.
Yeah, so as we were saying, we do this on a mass perspective, and so we can range anywhere from 0 to 100% by weight of drug and polymer. Now, if you look at again, as I was saying, if you look at the moles, though, you might have a 50% by mass weighted dispersion where it's half the mass is the drug and half the mass is the polymer. From a mole perspective, that's going to be about 99% of the drug in terms of the moles of drug is going to be, the molecules of drug will probably be between 90 and 99% in terms of moles. And that's what I mean by everything squishes up to the right, because the polymer is so large relative to the drug that it doesn't make sense to do things on a mole basis like you would see, say, if you look at ethanol and water, where you do see a lot of these thermodynamic curves on a mole basis. We don't do it that way. We do it on a mass basis because that allows us to actually do the math properly and to actually derive the derivatives of the slopes and everything that makes sense, because otherwise you get really strange results if you do it on a mole ratio.
Okay, thank you. Does Patheon have any publications available on this topic?
Want to take that, Tom?
Yes, we have published 3 different articles in drug development and delivery over the past 2 years, and we can provide those references. We've also presented this material at AAPS in 2015, and we will have a poster coming up in November at AAPS.
Mm-hmm. Yeah, I'd just like to note to everyone also that this presentation will be available on demand in the next day or so you can come back in and review it again. And now on to our next question. Can you accurately predict thermochemical properties such as glass transition temperature and melting point?
Yeah, we can, and in fact, we've done that with glass transition temperature, and as Tom alluded to, we've presented that at an AAPS poster last year, where we looked at for pure materials, we looked at the glass transition temperatures. Now we believe that we would easily be able to take this into dispersions of homogeneous mixtures of materials as well. And again, we don't want to just focus on solid dispersions. We can also look at lipid formulations and other formulations and predict those thermochemical properties as well from those.
Okay. Here's another question. How long did the calculations take?
So for the large, I'll answer the first question. For the large scale simulations where we have tens of thousands to hundreds of thousands of atoms, they can take anywhere from 48 to 96 hours, depending on, really, the limiting factor is the number of atoms in the simulation. And so if you've got a 200,000 atom simulation, that can take upwards of 96 hours, which is about 4 days. If you've got, fewer than 100,000 atoms in a simulation, and that all has to do with the amount of polymer and drug that are actually included in the system. The more drugs in the system, the fewer actual atoms, because drugs are smaller than polymers. You actually see a decrease in the amount of time. Now we run these on, as I mentioned earlier, on Amazon Web Services cloud platform, which allows us access to high-performance Linux computers with high-performance CPUs. But even so, because of the nature of the MD simulations, they still take a while to do. And some of the research that I'm working on is improving the speeds of these things so that we can get better turnaround for our clients.
Okay, great. Is it possible to simulate multi-component systems such as an API, a polymer, and a surfactant?
Yes, actually.
Yes.
Oh, go ahead, Tom.
Yes, it is, and that's one of the nice things that we can do with these calculations is look at multi-component systems. So we can look at mixtures of different polymers. We can add in surfactants. We can look at lipid surfactant interactions, polymer, different drugs mixtures with given excipients. So that's really one of the nice things that we can do with these simulations.
Right. Just sort of to extend on that, so can these methods be used with other formulation areas such as lipids, and can you perhaps give an example?
Yeah, they can, and in fact, we've looked at that for a number of different lipid excipients and surfactants like soybean oil, tween 80, et cetera, and we've got different models that help us understand what the solubility of drug in excipient actually is, and so that tells us whether or not we can get a loading factor that's appropriate for lipid formulation, which can give you a go, no-go decision on whether or not a lipid is the right choice going forward. And we run these MD simulations as well on these lipid excipients. It's similar in nature to the solids, although things move around a lot more, as you would expect, because these are actually liquids. Soybean oil is actually a liquid, and the drug is more free to move around. But we can derive the same sorts of information from whatever material we are confronted with. We can do these types of simulations and understand what the thermodynamics actually are.
How are the polymers chosen?
You want to take that, Tom?
Yeah, the polymers that we will start off with a list of 20 different polymers or so, and when we want to first start these things, we're looking for stability of a solid dispersion. So we look at things, interaction energies. How well does the drug interact with the polymer? And these are intermolecular forces. There's not covalent bonding or anything like that going on between the drug and the polymer. So what we do is sum up the different interaction energies, and those that provide the most stable interaction energy typically are chosen to move on in a development program.
Okay. Again, can you talk about the software you use for the 2D and 3D descriptor calculation?
Go ahead with that, Matt.
Oh, for 2D, I think that's yours, Tom.
Okay.
For the descriptors,
we have a suite of different programs that we have put together and compiled. So we calculate all kinds of different descriptors from a variety of different programs.
Okay, great. What is the percentage of success for prediction versus the actual solubility using this model?
You want to take that too, Tom?
Yeah. What we find is we are predicting typically over 75%, and that is compared with an experimental value. So we will take our candidate polymers, they will advance in an experimental program, and typically 75% of those, 75% of the time, we've accurately predicted which excipient will perform better.
Mm-hmm. And in terms of solubility, again, we're moving in the direction of understanding the dissolution performance, so that we can actually predict dissolution performance directly, which would essentially tell us what the solubility enhancement is of a drug molecule from a dispersion. And then, as you saw in the slides that I showed, in terms of the actual thermodynamic solubility of the drug in the polymer in a solid solution, we've got that fairly accurately as well. I would say that we're above 70% accuracy there too. 70% of the time we get that right. And I did show one nice example of nifedipine and PVP where you saw very similar energy values between the nifedipine and PVP thermodynamic mixing, the enthalpy of mixing, and that we predicted using the pairwise energy. There's probably additional energy we could extract out of the simulation that has to do with pressure and volume. That is probably why we don't see the exact same energy, is because we're missing one of those components into the enthalpy. But in any event, we're trending in the exact direction that we know we need to.
Okay. Let's have one final question. Can phase behavior be taken into account in your simulation?
Phase behavior. So we've not looked at that explicitly. And I think what we're mostly trying to understand at this point is stability and dissolution. But the long-term goal would be to understand, is there a way to extract from this from the energy and from the agglomeration, because we can actually look at the distribution of distances between drug molecules in a dispersion. And can that actually be correlated with known phase transitions, in other words, instability over time? I don't think that it's possible to simulate phase transitions because you're talking about something that's on the order of days, weeks, even months and years versus nanosecond simulations. But I do think that if you can extract information out of the simulations that give you a metric of what the behavior or what the actual physical state of the dispersion is at time zero, that might actually be predictive of phase behavior over a longer period of time. But that's not something that we would simulate directly.
All right, great. Tom and Matt, thank you very much for your presentations and your questions. I want to thank the audience for submitting their questions and participating today. If we didn't get to your question, it will be followed up via email. And I'd like to thank our sponsor, Patheon, for making today's webcast possible. Today's webcast will be available for on-demand viewing through September of 2017. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and have a great day.